Well, we have one of our own today, Juan, Professor of Engineering. Aren't you also a dean or associate dean or vice chairman or something?
Yeah, something.
You are.
No,
All right.
like that. Not much like that. I'm a vice chair of the department.
Why don't you tell us about CRISPRs?
Sure, yeah.
Okay.
All right, so my name is Juan Santiago. I'm a member of the mechanical engineering department. I work on microfluidics. And a couple of years ago, we became interested. Microfluidics are small devices that are used for bioassays, including things like DNA detection. And a couple of years ago, right at the start of the pandemic, we became interested in CRISPR, which is a very famous and well-used discovery and enzyme. And so I'd like to tell you about this. And basically what we discovered is a very strong pattern of misconduct by scientists. We try to point it out, and the problem is still just growing and getting worse. Even if you're not interested in biology or CRISPR or assays, I think it's a very nice case study of how how dissension is suppressed, maybe even punished, but certainly suppressed in science.
Hey, John.
Yeah.
Are you free now?
I'm free.
Okay, great. So
You got to turn your microphone off. That's Benny Elimelech from Yale. Okay, so the title's about CRISPR kinetics. It also applies to CRISPR diagnostics. I'll introduce you to this. I put here as my first author, my former student, Ashwin Ramachandran. I want to really highlight his contributions. He is a very brave student who basically did measurements in the laboratory which disagreed with Nobel Prize winners, with all of the existing papers published to date on the subject, and had the courage to sort of stick by his guns. Ashwin is now an assistant professor in Purdue. So I'll first introduce you to CRISPR, how we got into this field, and then I'm going to describe this catastrophe in publications. So CRISPR evolved from the immune system of bacteria. It was a discovery, part of nature. Something like half of the strains of bacteria have some sort of CRISPR system. CRISPR itself is a nested acronym. So it stands for clustered regular interspace short palindromic repeats, and then Cas is used, and Cas is the nested acronym. So the C in Cas stands for CRISPR, and CRISPR stands for that. There are two major branches or forms of relevance of the discovery of CRISPR. The first one is gene editing. So gene editing, and I think this is really the more important one, but it's become a way that you can knock out genes in an organism, in cells, but also in the hope is in humans as genetic treatments. You can also use it to add genes, etc. So alter the genome in an easy way. There was ways to do this before CRISPR, but there are now much more powerful ways to do it with CRISPR. The second branch, which has received a lot of investment and startup company interest, and during the pandemic really ballooned, is CRISPR's role in molecular diagnostics. So that was first shown a few years ago. I would say the first detection of SARS was early in the pandemic. So that was this one of these two big companies in CRISPR, this one called Mammoth Biosciences. And of course, there was a Nobel Prize in CRISPR in 2020 to Jennifer Doudna of Berkeley and Emmanuel Charpentier of Max Planck. So I'm going to be talking about this. This whole talk is about how to use CRISPR to do specific detection of genes and how to use it to do molecular diagnostics to detect things like cancer, but also pathogens. So the way these assays work, maybe I'll stand over here. You start with some sort of sample. It could be blood, urine, could be a nasal swab, cheek swab. And the target may be genetic material indicative of cancer. It may be microorganisms and pathogens. So the yellow blob here is my schematic for CRISPR. CRISPR's functionalized with a piece of synthetic RNA. So the synthetic RNA automatically acquires a three-dimensional shape, which has an affinity to the enzyme itself. And that's this little hairpin schematic here. It also includes a tail, and this tail is then complementary to the target of interest. So when you mix it in with target, this complex, the guide RNA plus the CRISPR, will find the target. For example, say the target is double-stranded DNA in the case of Cas12a. It can melt it, it can open it up, detect the complementary strand, and then it makes a cut, okay? It cuts at that specific gene location. And the way that you make this into a detection method is once this happened, a little piece of the target stays in the CRISPR. And I think of it as a finger that's now on the trigger of CRISPR. So thereafter, CRISPR is what's called promiscuous. It'll go on and cleave all DNA, irrespective of sequence, okay? So you turn it on here, and this is a specific reaction. It's also called the cis cleavage. And then you detect it by leveraging the so-called trans cleavage, which is this promiscuous activity that it acquires, and it starts to cleave things. Cleave any nucleic acids. In the case of Cas12, it's DNA. In the case of Cas13, it's RNA, another CRISPR ortholog. So here in this picture, I'm showing the most common way of detecting, where you add probes. So these are fluorophore quencher synthetic probes. These are single-stranded DNA with a fluorophore on one side, a quencher on the other, and there's a near-field interaction. So when it would emit a photon, it gets quenched by the quencher, and much less fluorescence happens. So this creates a fluorescent signal. This is real data right here from our lab, where we're increasing the concentration of the substrate here of the enzyme being these probes, and then we're detecting this fluorescent signal. The most successful enzyme kinetic model of all time is this Michaelis-Menten model, and it's also the most successful in describing CRISPR's kinetics. But this is describing here the trans cleavage. So this Michaelis-Menten is used for the trans cleavage. So here we have the activated enzyme. Activated enzyme shown in blue. It's promiscuous now. It's cleaving these reporters, and it does so by first picking it up. It can disassociate, or it can cleave and create this fluorescent signal. There's a regime that's applicable to CRISPR of the velocity of this reaction, which has to do with two kinetic constants, and I'll describe these, but one is called the kcat, which is when the substrate is abundant, it's the maximum amount that you can turn over these reporters. And this km, which describes basically the net affinity for this. It's the off rate versus on rate for the state of the complex.
Just don't spend a lot of time on this, but
yeah,
what's the object? The object is the little hairpin thing.
Yeah. So the object is you want to recognize a specific sequence in the target, which came from here.
Yeah.
Right. And this little cut, you can't detect it by itself. So you use it to transform this enzyme to now make signal for you. And you detect the signal.
Just briefly, you attach something to something you're interested in that has a signature, and then you have something else that can tell when that signature is.
Yeah, pretty much. So, and the really nice thing, I'm going to criticize the scheme, but the really nice thing about this scheme is that, so I can send an email and get synthetic RNA within a couple days. So now I can reconfigure this. So if there's some new pathogen, within 2 days, I can mix it with the CRISPR. Now I'm detecting this new pathogen. So this part is a synthetic manner in which you achieve highly specific, and it is highly specific, detection of a specific gene.
Off-target guide RNA, that this is something that, this is what you're tested, you're trying to pick up, whether it's cutting the wrong thing, is that?
So there's a few things wrong. Yeah, so one of the things that is wrong, and it's actually pretty good about this. So one of the things wrong with this scheme is that if you have, say, point mutations in the target relative to what you thought it was, CRISPR will still detect it, right? Especially 1 or 2 bases out of 20, it'll still detect it. Another thing that's wrong with this is it turns out that in the absence of target, CRISPR starts cutting as well. Okay? It starts cutting these reporters, which is a false positive. Okay? It's a low rate, but it's a measurable rate. And hardly anyone talks about it, even after thousands of papers. Okay? The second thing wrong with this is that these quenchers aren't perfect. And the cleaved to uncleaved fluorescence intensity ratio is only like a factor of 20. Okay, so that limits your dynamic range. You have low fluorescence, and then you have higher fluorescence, right? The third thing that's unlike other things like PCR, this is a linear amplification. So you weigh twice as much, and you get twice the signal, right? You weigh 3 times as much, 3 times the signal. There's no exponential amplification of this. Okay. Any other questions? Yeah, please.
I guess, is it, the thing about the 2 categories of editing and diagnostics?
Yeah.
Does your findings in diagnostics have any spillover to the editing literature? Or do you see that as more on solid ground?
No, the editing, I think it's very good. And it turns out that the kinetics, well, the off-target issues definitely apply to editing. And that's still the remaining challenge. And I'll talk a little about specificity and so on. But the editing that's typically done, they have tens of hours to accomplish it. They drive it with high concentrations of CRISPR, and the kinetics don't become as important. So they just want to know if they edit or not, and if they edit the right gene or not. And the rate and the signal and all these things don't matter. So my talk really is criticizing the kinetics literature and the diagnostics literature of CRISPR, which is a large part of the investments and the interest in CRISPR. And I'll show you that, it's say 1,000 papers a year now.
Is that of equal importance to the editing?
Sorry?
Is that of equal importance to the editing literature?
I think the editing long-term is more important. And I think that that's the major reason the Nobel Prize was conveyed. And this diagnostic thing seemed like a fast way to get revenue out of the thing.
Yeah, sorry, can you repeat?
Sorry?
Fast way to?
To make money out of it.
Okay.
Yeah. So here I'm summarizing what I consider the 2 seminal papers in this CRISPR kinetics and CRISPR diagnostics field. On the left is work from Berkeley. This is Jennifer Doudna's lab. She's the Nobel Prize winner from 2020. In a science paper, they first identified the scheme that I'm talking about. So all this stuff is pretty new. 2018, they first identified that after it sees this highly specific cis cleavage, the enzyme becomes activated, and this activation unleashes here indiscriminate single-stranded DNA activity. So then it starts to cleave, and hey, we can use this as a detector. One of the amazing things in this paper is they found the KCAT. This is the amount of reporters you can cleave per second, and it's exactly what you would think it is. So the maximum amount would be about 1,200 reporter molecules per second that it can cleave after it's activated. They concluded that the enzyme is a diffusion-limited enzyme, which is a special class of enzyme which can act so fast to make these reactions that it's only limited by the diffusion of substrate as it approaches the enzyme. Okay, kind of rare for such a thing, especially rare for DNases, for endonucleases, et cetera. So they reported this 1,200 turners per second. Shortly after, just a few months after, The group of Feng Zhang, so this is Feng Zhang from MIT. He's one of the most famous labs. His is one of the most famous labs in CRISPR, both for diagnostics as well as gene editing. They published in Cell Reports the second paper ever to measure this effect, this kinetic signal producing effect. They measured it for Cas13b, which is an ortholog. That 13 means that the CRISPR, the cis cleavage, targets RNA. Okay, this turns out to be a big deal, right? Because if this is sensitive enough, if these kinetic rates are true, maybe we can detect the virus, the COVID virus, directly, right? That was became very relevant shortly after this. But things like the flu, for example, we can detect it. And they reported about 1,000 turnovers per second. Now, these are two sort of major labs in the field, and I think their reputations, their status in the field really led to what I call a catastrophe, where people started publishing complete garbage all over the world, and are still doing it today. Okay? So we'll get to that. I want to sort of remind you, so Doudna here is the Nobel Prize winner. This is, I'm just going to play a few seconds of it, but a clip from her Nobel
It
Prize.
also triggered our interest in the biochemical activities of other families of CRISPR-Cas proteins. And in research done—
She's describing this paper here on the left.
Janice Chen,
Nobel Prize lecture on CRISPR.
A family of enzymes called Cas12 also have the ability to cleave single-stranded molecules, in this case, single-stranded DNA, upon recognition of a double-stranded DNA target. And in this experiment, what Janice Chen showed is that the Cas12—
Janice Chen, her postdoc, the first author.
Double-stranded DNA
Okay, I'm going to pause there.
also triggered our interest in the biochemical—
Maybe I'll keep going. Sorry. Okay. So she said that triggered our interest in this biochemical assays and using this as a medical or a diagnostic for pathogens and things like cancer, et cetera. A couple of the authors of this are now officers in a company called Mammoth Biosciences, who's been very successful, raising hundreds of millions of dollars. Much of which, and the fanfare around the company, was around using this for diagnostics, quick, sensitive diagnostics. So they recognize that the gene editing would take longer. It's harder to break that into a market. You're changing people's genes. That's a lot of work has to be done before that's safe and more applicable, more generally applicable. Okay. I also mentioned that a couple of the authors here are now officers in a company called Sherlock Biosciences, which is also focused on the CRISPR. Their first products are just being released, and their first products are all around this molecular diagnostics. The Feng Zhang himself, basically the owners of the patents here, which is like the Broad Institute on the right-hand side and Berkeley on the left-hand side, they've now fought in court about this. So despite the fact that Doudna received the Nobel Prize for discovering these mechanisms of CRISPR, the jury in the trial found that, in fact, they had not sufficiently enabled the invention. That is, they hadn't described how to do the invention in their own patent. And they ruled that, in fact, Feng Zhang had done this before, okay? So very high profile in this world, the fact that they now have this win in court over who invented this type of technology. And I'm going to talk about both of these companies later on. But just to give you a summary of some of the trends, these marketing companies in biotech project something like, for this year, $2 billion with a rapid growth rate. Although most of the projections into the future from today are in gene editing. If you go back even a couple of years ago, a lot of the hype and investments were around diagnostics. So this second bullet is about the diagnostics. There's been, just from a few anecdotes that I've put together, something like over $1 billion of investments in startup companies just for the diagnostic component, okay? Just for the diagnostic component. I'll give you a reference for that. These here are all really focusing on the diagnostics and the kinetics and so on. Yeah. Oh, please.
So, you said that Sherlock is a product.
Sherlock is about to release a product. It's not already released.
What is a product in this world?
Oh, so it's a kit to, and I think their first application is going to be for a take-home kit for sexually transmitted diseases. We're going to go on a date, and I'm going to go to CVS and say, hey, I have no chlamydia. I'll pick you up at 7.
Mhm.
So that's the kind of their business plan.
Doing
I
right
wasn't
in front
there. Sorry?
Do it right there in front of them.
Yeah, I want to do it in front of them. That's right. Or at least my cousin, who gave me some cheek swab. Oh, no, blood, I guess.
So
Okay.
like complete innovations, there's no other product like them?
Sorry?
These are completely new innovative products, there's no other type of tests like that exists currently?
No, there's other ways. Their claimed value added is the convenience. It's a one-pot assay where they're gonna have to amplify, then do this CRISPR detection. And I think it works. I think it'll work. They originally talked about doing it without amplification, which I think is impossible. Okay. Without pre-amplification. I want to tell you how we got into CRISPR, and I just have 2 slides of my own research, because I don't really work so much in CRISPR, not anymore actually. But we work on microfluidics, little chips like this. I'm going to spend about 30 seconds on this. We use electric fields to pre-concentrate reagents of many reactions into little shock waves. So this is like an 80 micron wide channel. We're going between 2 filter fluorescence cubes. The red indicates the presence of the CRISPR enzyme, and the green are the reporters that it's cleaving, because it's now detected some amount of target from a cheek swab of someone who had COVID-19. We got this assay and submitted this paper by about late May, early June of 2020. It's still one of the fastest ways to do detection of the genetic RNA. We can do it in about 30 minutes. But what we found when we got into this, so let me go back here just to this slide. We're good at using these electric fields to achieve pre-concentration of reagents inside this little shock waves by up to 10,000-fold. And that can translate to 10,000-fold increase in the reaction rate, not the kinetic constants, but the reaction rate, right? We know how to make reactions go fast, and we use these electric fields. So when we first got into this, we looked at all the data, all the publications of the kinetic rates. We took this number, we did back of the envelope calculations, and we said, wow, we can detect CRISPR directly. We don't need amplification. We'll just be able to do this. And we spent most of that time from February to May of 2020 trying to do that, right? And we failed. So in the end, we had to use the electric field in 2 steps with a manual step in between, which we don't like. So we did a nasal pharyngeal swabs. We can also do cheek swabs. We put it into our chip. We use this electric field to extract and purify the nucleic acid. Then we do reverse transcription and LAMP. LAMP is an amplification method that we didn't want to use, we didn't invent, but it turns out we needed to use. And it's very convenient. LAMP is isothermal. Its temperature's not too high. But we did this in a standard thermocycler, and then we go back into the chip and we speed up the CRISPR reaction. And we're able to do this with no false positives, speaking to the very good specificity. And we had just 2 false negatives for all these patients that, according to the WHO RT-PCR test, were positive for COVID. These 2 that were false negatives had very low counts. So their CT, if you're familiar with PCR CT, their cycles of amplification were like 34 or something. They're really high, really low concentration. Okay. So why did we need pre-amplification? After we submitted and published this, we went back and started looking more closely. We made our own model. So the way people determine these kinetic rates, this Kcat for the maximum rate and this Km. The way people determine this is they put in the activated enzyme with increasing levels of reporter. They focus on the transcleavage because the transcleavage is the rate limiting step, not the cis cleavage. So they focus on the transcleavage. You put in increasing levels of the reporter, you measure kinetic rate for each one, and then you plot these reaction rates as a function of the substrate concentration. And then use various theories, most notably this Michaelis-Menten. We did this full, it took some coupled ODEs, very easy nonlinear ODEs to solve. So people then fit this in order to extract these two parameters. And so we could do this as well. And when we measured these kinetic rates in our own lab, we would get, this is the best one that we measured, was about 3 turnovers per second, not 1,200 turnovers per second. And if it is that slow, this is why we can't do it. So the next step that we did is we said, okay, let's look at, for example, the Dudna paper. It's the most cited paper. It has like almost 4,000 citations, even though it's just a couple years old. Let's look at this paper. We're going to look at the Kcat and the Km, the kinetic rates that they reported. We're going to put them in our model, and we're going to predict what their raw data should look like. Okay? So we're looking for consistency. We're using their rates comparing to their raw data. Nothing with our lab. So this is their raw data, and this is our model with their kinetic rates reported. Right? This is not a good fit. Right at the initial.
Just so I understand,
Yeah.
the model isn't your model, it's taken off the existing models, right?
Exactly. It's the same model they claim to use.
But
So
that's
we
what I'm
use their
asking.
model
Yes,
with their kinetic rates
Okay.
and they're comparing to their raw data.
Okay.
Yeah, so we're looking for self-consistency in their papers.
that's right, okay,
And all of this is independent of measurements in my lab.
yeah.
Did they have a type of figure like this?
Sorry?
Did they have a type of figure like this that showed a good fit?
Oh, their figures have curves with circles over them.
I guess, is there like,
Let me tell you what happened.
okay.
Okay. It was completely wrong, complete garbage. It's complete garbage, okay? That's the short answer, but I'm gonna try to show you. So this is the predictions using their own Kcat and Km versus the model. And I want to say there's interesting time scale, which is the linear time, the time you spend in the linear regime where this signal is increasing linearly. And so here from these, you might estimate the linear time scale here is something like 10 seconds, but according to their raw data, it's greater than 400 seconds. So that's 10 seconds versus 400 seconds. Okay. We then started doing this with every single CRISPR kinetics paper ever published at the time, which were 9 papers. And we got the same sort of result. Okay? Their own rates don't match their own raw data. And they all sort of support each other. Everybody gets around 1,000. Some people,
This is just a quiet conspiracy.
let me tell you what else we did.
So this is also true for the MIT group.
Oh, yeah.
The other.
Absolutely. And groups in China, groups in Europe, everyone published it. Let me show you what we did. So to analyze and present this discrepancy within each of all these papers all over the world, we define these 3 non-dimensional numbers. We're engineers. We like this sort of approach. These are, we call them alpha, beta, and gamma. There are 3 numbers that if your data is self-consistent, they all have to be less than 1. Okay? We don't invent the laws of physics and chemistry, we enforce them. So the first one's pretty simple. The time you spend in that linear regime, right? As the signal's increasing, it first goes linearly and then it saturates as the first order exponential. The time you spend in the linear regime times the velocity of the reaction, the maximum velocity of the reaction, is the number of reporters you've cleaved. That should be less than the reporters that went into your tube. It's conservation of mass. We're big on it,
Yeah.
right? This one is the maximum velocity you claim to have observed should be less than the theoretical maximum velocity, according to kinetics theory. And the third one is the time you spend in the linear regime should be less than the time you spend in the entire reaction. Okay? The time I spend going to my car is less than going to my car plus the supermarket. Except here, the supermarket would be in the next town. Okay. We then compared this. You asked about those two seminal papers. Here's the Chen paper and here's the Slaymaker paper. So for example, the MIT group, they observed 10,000 more reporters than went into their tube. Okay. The Chen observed, they spent 70 times in this paper, in this measurement here, 70 times more in their linear regime than in the entire reaction. And then you see some amazing ones. So these people spent 70,000 more in their initial reaction than in the entire reaction. They observe, these people here observe, well, that was the record for 10,000. Here's another interesting
Is
paper.
Koski on your side?
Sorry?
Is Koski on your side?
Koski on my side.
Koski
Oh, this
is
paper.
fine.
Oh, yeah. So this is interesting. So Koski, one of the senior authors in Koski is also Dubna. And if you read that paper, what they did is they made changes to the guide RNA. And what they found is that the changes didn't work. It completely reduced the rates to very low values. And so they basically conclude by referring to their earlier paper and saying, those are the real rates. Forget what we're doing here. It didn't work out. Okay, so even this, I think, is not really a motivation. It's not really.
Why do they want these high rates? So you could do it without the high rates, but just amplify it. Why do these guys want all these high rates?
Oh, if you do the I rates, then you don't need amplification beforehand.
And that's the whole point, and somehow that's a really great thing.
It's a great thing because now it could be a very simple assay. Now we can do it at home.
I see.
It's also kind of sexier. I think people like that when they're publishing.
So
Yeah.
can I clarification?
Yeah.
So do they report alpha, beta, gamma in their?
They don't.
Oh, they
So
don't.
these are-
So these are calculations that you've done based on what they.
Exactly. But actually, they're very simple. So
Yeah.
this, this is the rate of increase in their raw data. This you can estimate. We really like, for example, this one and this one. Because even if you get your y-axis wrong, you just put random numbers there. I can tell linear timescale from your x-axis.
Yeah.
That's key. Yeah.
So
go ahead.
when we get to their motivations or
Yeah.
whether there's something fabricated,
Yeah.
are there not standard sanity checks in the literature? Like these three parameters? Or is it something
Yeah,
you innovated on to really examine it?
The Michaelis-Menten kinetics, they should be self-consistent. Your calculations should be self-consistent.
Right.
One of the
Why would the
excuses I've heard from groups is, we're not quantitative. We're not quantitative people, we're biochemists.
The authors say that?
Yeah.
Yeah.
What?
Sorry, go.
What
But they're
about
publishing, yeah.
But there are referees for papers, so of course.
Oh, yeah. Actually, it gets worse.
They can't redo the
The referee are
experiment.
not quantitative.
So let me keep going. So, oh, by the way, so these two, this is really interesting. Whoa, what happened here? These two hold the record for the highest kinetic rate ever measured for any CRISPR assay. These are two orthologs. One is cleaving or detecting RNA, the other is detecting DNA. This one is in China. This one is in Florida. They're measuring, they both have the record. One came right after the other. And one came significantly after the other, who refers to this one and says we didn't quite make it. They matched to 3 significant figures on a number that you can't measure within a factor of 2 or 3. You can't measure this within a factor of 2 or 3. But they both hold near record of each other, and it's just complete garbage. Okay, so first thing we did is we started emailing every one of these authors with slides showing, look, this is how we're measuring this alpha, beta, gamma, this is how we define it, we'd love to meet with you. Basically, no one answers, complete silence. We do this several times, we contact editors, complete silence, okay? No one answers. To their credit, Feng Zhang replied to one of my emails and said, hey, let's meet over Zoom. And we met, I don't know, an hour and a half. At the end of the hour, they went off, you know, in Zoom, you can have like a breakout session. Him and officers of the company, Sherlock, went out. And when they came back, they said, yeah, you guys are right. This is completely, we're off by a factor of 1,000. Okay, so to their credit, they did that. We then used this communication.
Did you have that recorded?
No. Sherlock
Okay, so
still-
therefore it's, this is just your gossip.
They ended up putting this in print.
Okay, they did put.
Yeah, I have
There's
that.
a record.
All right, yeah,
Okay,
sure. Oh,
Sherlock is still selling that product now.
Not that product, their product all now have amplification because it's impossible to do without amplification. Despite the fact that there's going to be 1,000 papers next year on. There's going to be 1,000 papers next year publishing impossible stuff. Let me tell you, I'll get to that in a second. So we try to do this. In the meantime, there's 5 new papers, all publishing complete garbage, all agreeing with each other. Like no one makes a mistake of 1,000 times too small. Everyone's 1,000 times too large. That's also suspicious, right? In the meantime, Dudna is awarded the Nobel Prize. She also finally, Janice Chen, her postdoc, finally contacts us. We tell them, hey, look, the MIT people agree. We think your first paper, the one that's most cited, is completely wrong. Let me tell you just a couple anecdotes. I'm not going to go through every little detail. But one of the first things we did is we contacted Science and we said, look, you have this paper that's touted thousands of times per year, or about 1,000 times a year. And we think it's completely wrong. Here we're showing that it's not consistent. So here's one of the emails I sent. This is senior editor Gemma Alderton at Science. We explain this. We point out that there's a big mistake in the 2 seminal papers in the entire field. We'd like to offer this to Science. Science supposedly encourages comments on published work in Science. Their response is, look, we'd rather deal with the authors. So after we submitted our confidential manuscript to Science, Science responds and says, no, we're not talking to them about it. And we'd rather have them correct it, not you guys. We don't know who you are. Okay? So be assured that we are in discussion with the authors of our paper.
Then it's a problem about time. Are people not understanding the consequences, the equilibrium consequences of the actions? Like that means nobody is going to be checking Science.
Oh, yeah, for sure.
I mean, and this is
This
is so ubiquitous in society.
There may be
You
others
make a
besides
decision
mine.
for your own benefit, right? And you ignore the externality.
But
But
this has been going on in economics for decades
I'm
and nobody in
confused.
economics cares.
I'm confused because I published in Science.
Yeah.
And Science, you could write directly without writing an email. You can write a comment, which they have to take into account. They can reject it.
We submitted several ways
So
and they
you-
wouldn't
you-
review it.
I see.
So did you try technical comment?
Yeah,
This is
exactly,
the-
yeah.
The type of paper that would be appropriate for this thing, you know, if a paper that they have.
We ended up
published is
publishing this.
flawed,
Yeah,
then a technical comment supposedly.
within Science, for Science, you're
For
talking.
science is trying to
Yeah.
correct the error.
We try.
While a perspective, you know, they would say a perspective is more like reviewing the field. as they
Absolutely.
say.
But they violated confidentiality rights.
So I'm just showing you one of the emails,
That's-
because it was the most clear, the response.
Yeah.
But we try to submit this any way whatsoever to Science, and they told us, we are not interested in your submission. We're already gonna fix it. We're working with the author who made the mistake. We don't need you to point it out. That's what they told me.
Yeah, that's, so all this
That's
other
why
stuff. I mean,
I think it's so outrageous, that's why I'm putting Gemma's name on this email.
But from her
Here's
perspective,
something that I think even more, sorry, go
was she
ahead.
trying to save face? Like what, thinking about her incentives, why would she rather have them?
Their author just got the Nobel Prize. Their author I don't know. That's
Rather than
maybe
starting
that's. I'm now
That's what I
criticizing.
get, my guess is, yeah.
We then try to go to archives. Archives wouldn't take our.
Oh, boy.
So our
So-
preprint, so here's the email from
So what
archive.
is bio,
So here's the only response we were able to get from archive.
Yeah.
We regret to inform you, your manuscript is inappropriate for bioarchive. Right? This is exactly what it's for. We're supposed to be able to put preprints here. We didn't pass their two-step review process. Everyone is examined by an affiliate scientist to determine suitability. Ours was not. We could not get any other explanation.
Question.
Yeah.
Very,
Have you ever heard of this? You're very,
Oh, yeah, yeah.
people getting rejected from archive.
I have been rejected.
Oh my gosh, okay.
Very similar.
Yeah.
Technically, was the formatting of what you submitted,
Yeah.
like introduction, methods, results, discussion, and then abstract? Because if it's not,
Yeah, we had an abstract.
scientists may not even be scientists. There may be bots that just look, for structure, and they say, well,
I
this
see.
looks more like a comment rather than a paper.
Yeah,
And they may reject it on that, ridiculous. But if you format it like, standard IMRAD, introduction, methods, results, discussion, it's very difficult for them to.
They do it anyway.
it was a two-page manuscript. It had an abstract, it had an intro. The intro was short, it was like
Yeah.
a paragraph. We just talked about the two papers. Then we went alpha, beta, gamma. We're drawing.
Did you have separate methods and results and?
Yeah, we try to follow as well as possible. I don't know about separate methods. I think we went from introduction right to discussion to conclusion. So I think we had 3
Maybe
subsections.
that's,
It could be
it is
maybe
ridiculous that
Yeah.
we do this, but it does happen. And
Okay,
about 30% of the submissions in bioarchive and meta-archive are.
don't make it through this. Oh, interesting, okay.
The science editor you mentioned
Yeah.
is now consulting as a freelance editor writer for researchers in biological sciences at a company called BioSci. And after 18 years at top tier journals, it says no longer doing that. And she's also, quote, writing a careers talk about what I have learned after 18 years as an editor at Nature Publishing Group and Science. And asks actually on X, if you were going to my talk, what would you want to know? So maybe you could
Okay, good.
line on that.
Okay, so in the meantime, we expanded our analysis. We submitted, I won't take you through all of them, but all of them will not go to review. We tried the Cell, because Slaymaker had published there. We tried Nature Biotech, Nature Com, I think those 3. I think we also sent a couple informal emails and people, well, we're not interested in this type of stuff because you have no new data. And we had like no new data on purpose because we're trying to show inconsistency in previous data. Okay, in the meantime, Jennifer Chen, the postdoc worked for Dudna, and Zhang, and who's Slaymaker, who's on the call, they ended up saying, we told them, look, we're posting this on our website. We encourage you to also post a correction. They published the correction.
it was accurate?
Yeah, so here's their, well, actually, it was a correction in the right direction. I think Zhang's is more accurate than Dudna's correction. So here they're saying, okay, we said it was 1,200. We now think it's 17. And here they said, look, we said it was 1,000. We think it's less than 1.
Wait, on the student, what's the explanation for the error? I mean, that's a pretty big mistake.
Great question.
Wait, you got
Both
reference
sides of
for,
this. On
what?
both sides of this. So here say, hey, this error was pointed out by Santiago. This one was kind enough to also include my student, Ashwin. On the left, Chen made a units error, and Slaymaker made a units conversion error. Okay, so on the left, you put these reaction vessels into a thermocycler, you close a box, it goes up to a certain temperature and it measures fluorescence versus time. The fluorescence comes out as arbitrary units, right? And then it's up to you to calibrate your instrument to put in fluorescence of known concentrations and measure the signal. Now you relate arbitrary. Chen set arbitrary units equal to micromolar. That was the mistake she admitted. Right? Arbitrary units equals micromolar.
Yeah, okay, right. She didn't get it.
Eventually inhomogeneous, this equation, yeah. Slaymaker made two mistakes. One was seconds to minutes. Okay? And I believe these were two honest mistakes in the first two papers. Seconds to minutes, and the second, I think, was he forgot like a 15-fold dilution. Or, sorry, included a 15-fold dilution that never occurred. You multiply that and you get about 1,000. Our reviews finally come back. So Analytical Chemistry finally agrees to review our manuscript that's now published. The reviews come back and they're pretty harsh. Like, this is crazy, you guys can't put this out. But in response to that review, we now say, hey, look, the two seminal papers now agree with us, not with the reviewers. So the editor ended up going to publication and published it. And let me just tell you a couple, these are just fun stories I put in for color. DARPA, all the way from 2019 to 21, had two programs just around CRISPR diagnostics, CRISPR kinetics. They became COVID detection programs. The larger of the two was at least $70 million. By the program manager was Commander Dr. Jean-Paul Chrétien. He ignored my emails about it for 6 months. My emails were saying, hey, all of the money that you're granting all of these people, oh, by the way, in order to get any money from his program, you had to do detection without amplification. That was a condition. And many of those papers that I showed you, they all cite his funding. Do you think he'd be interested in this?
There is someone with
Oh yeah, please, go ahead, Eric.
Oh, yes. I'm interested in the preprint culture of this field. I'm in economics, and there nothing ever gets rejected, and you can put things up on your own website is common too. So it sounds like there's at least a formatting thing here. I was wondering, did you put it up on the web somewhere? The previous slide cited something in the archive also. So what exactly was the and did you distribute it?
We never put anything in the two major archives, which at least in these fields are archive and bioarchive. We were trying to move as fast as possible. In the end, our observations are super simple. They're based on three algebraic equations.
It's a theory.
Right? What's that?
It's a theory. You're not using data, you're taking their data.
I'm taking their data, comparing it to their other.
You need to re-register.
Exactly. I'm comparing their figure 3 to their figure 5, or their table 5.
Now, does it matter that it wasn't in those archives? If you just put it up on your own website, is that not just as good? And send 20 copies to your friends?
Oh, no, I doubt it. I can put anything in my website. In fact, after all this is done, we've published now 7 papers pointing this out. And most people completely ignore it and are still publishing garbage.
Even though the original people corrected stuff.
There are papers being published by the original people who cite the uncorrected work.
Have you tried actually just sending it to 100 people? Because one thing, I'm an old economist now, and I noticed that publishing is not good enough. You actually have to send the paper to people too to get them to read it usually, in our field.
Yeah, but we're now talking about 1,000 papers a year.
Okay, so let's see.
Anyway, ignored my email for 6 months. Finally, I had funding from another DARPA manager who urged this DARPA manager to take my call or at least listen and read the information. By then, we had published it. So this is October 2021. Chrétien claimed that he could not Zoom with someone outside of his program, that any video conferencing was impossible. He agreed to a voice-only call. There was a bunch of attendees on the call. He wouldn't introduce them.
Well, why did you care about this guy? Let him screw up with the...
Just an anecdote. I'm just saying, I don't know, maybe you care. This guy threw away $70 million a year of tax
So you're
dollars.
worried about the fact. He's probably not
Talk
the
to
only one.
Elon Musk after January 20th about wasting government.
That's right, maybe Doge will address this. Here's another interesting one. So I met with a member of the Mammoth Biosciences Board, to whom I was introduced, on the condition of anonymity. I'm not going to say his name. Person expressed shock, seemed convinced. Absolutely no follow. After that, wouldn't answer emails.
Right?
So basically, I'm saying, look, there's the first products that you're promising within a year from your company are impossible. It cannot be done. Okay? And you've raised hundreds of millions of dollars promising these diagnostic, not gene editing, but diagnostic products.
Juan, you have another question?
Oh, please.
From Eric.
But actually, I'll just put this up.
Let him finish.
So
Okay.
a little over a year later, Mammoth completely lays off their entire diagnostics division. So maybe that was the feedback. There was no email feedback, but I read this later. Sorry, what was the other question?
No, let's go. Finish what you're doing. I'm in charge here. Finish what, I want you to finish because you're supposed to, we're already
Okay.
50 minutes in. Okay.
Gotcha. All right, so some follow-up. So we published 7 other papers on this. One of them was showing that the typical kinetic rates are on the order of 1. We looked at 7 of the most popular orthologs. That's a huge amount of work. We think that the background activity limits you to about 1 picomolar range, which is 1 million attomolar. Yet now the new problem in CRISPR is the following. So I'm just going to go through this kind of quickly. CRISPR is very specific, and we've shown that, and others are now leveraging that. I published a perspective eventually on this 2 years ago, where I made recommendations, things like people should share the raw data if asked. And none of that happens, by the way. Just to review some of the things that are happening up to now, the biggest trend is the number of kinetic rates publications is increasing, but people now don't provide enough data for you to check alpha, beta, gamma.
Yeah.
When they don't provide it, they usually get fantastic rates. They're amazing, fantastic in the sense they're fantasies.
So how are they doing it? Because it's not a common down, they're not making a mistake. They're literally saying, well, something in economics, whatever, I'm filing my own thing. Shut up, John.
In economics, we're gonna talk about-
Then what we do is we just say,
No, you don't.
When they do publish enough data for us to check alpha, beta, gamma, then their rates are really low. Okay? Sorry, almost always their rates are really low. When they publish enough for us to check alpha, beta, gamma, like this one, and we can, and their rates are high, then their rates are garbage. So basically, most people now don't give you enough information. And if they do, the alpha, beta, gamma is a great way. And if it's your own data, it should take you 10 minutes to take these 3 ratios.
The lining is that they've read your paper.
That might be, they maybe all read our paper, yeah. They certainly don't cite it, but they maybe all did. And here's us trying to, these people are pretty prolific. This is us trying to get data from an author who has signed a document to their editor saying they will share any raw data if you get asked for it. Okay? So look, this is maybe the 3rd email we've sent. Hey, we'd like your raw data. Look, here's the place where you sign. Authors must be willing to distribute their materials.
Is this you?
Yeah, this is me quoting the journal to them. I eventually sent emails to the editor. Editor doesn't help. Right? Editor won't. And this is iScience, not a bat from Cell Press. Hey, it turns out he and I had been in some DARPA program together. Hey, great to hear from you. Any word, a couple of weeks later, any word in sharing, please respond. And then one of the last responses, our legal counsel has been.
Yeah.
That's the, you forward this to the editor doesn't care. 12 emails.
Legal
Actually-
counsel of the university?
In the end, we got an email from them where they said, hey, we discovered that we had written down something wrong in our raw data. Now that we correct it, it exactly goes just under alpha, beta, gamma are just below 1 now.
Oh.
So they went from a couple hundred beta gamma to just below 1. Yeah. So what's going on more currently, this field is growing, I'll just say that. And here's maybe a summary. So these are not cumulative, these are rates. So CRISPR diagnostics, not the gene editing part, but just the diagnostic part is something like 5,000 papers per year currently. Almost none of them cite this catastrophe as if it never happened, and are still publishing rates that are 1,000 times off, okay? Most of the data, including in the journal where we published our paper with the same editor, okay? Amplification free, so this is what we think is definitely impossible, right? If you do amplification, you can do this, but we think this is impossible. We're headed toward about 1,000 papers a year. Almost all of them, at least 900 of these, I think, are impossible.
And the blue one you think is more possible?
No. Blue one is all diagnostics, but many of these now do
Including
amplification.
the red.
If you publish real stuff, you have to amplify before you do this detection. And so you're only using CRISPR for the specificity, not the sensitivity. This is one of the abusers of diagnostics. This is a paper from a group in Berkeley. It includes Dudna, but the senior communicating author is a couple other profs. But just to show you, during the pandemic, they took this nasal swab, they mix it in a little chamber, just straight with the Cas13, and then they put it over a phone, and a 2-year-old iPhone, and they can detect the virus, the COVID virus. The reported limit of detection is 100 attomolar, 0.1 femtomolar. That's about 100 copies per microliter, right? That rivals PCR in this field. This is about LOD for PCR from nasal swabs in COVID. We estimate, because we've studied the exact same enzyme with the exact same reporters, their signal is something like, that they're claiming is signal, is 1 million times weaker than the background signal, than if you have no target. If there's no COVID in the room, this enzyme produces a signal that's 1 million times stronger than the one they're claiming to detect with a phone. Okay. We've communicated to them, no effect. They won't publish anything, they won't agree to look at it. They do in their paper cite 600 turnovers per second and compare it to Slaymaker. They're comparing to the uncorrected version.
So why do you say fantastic paper?
Yeah, it's fantastic because it's a fantasy. It's like, unreal. Yeah, maybe I should put it in quotes.
So none of these, is some of these being commercialized? No.
With amplification only. It only works with amplification. Without amplification, you can publish anything you want. You can detect anything you want. It's just garbage. It's just a lie. This is Jennifer Dudna talking about this specific paper.
SARS-CoV-2 pandemic that has become a worldwide human health issue. We and other SARS-CoV.
Anyway, I won't play it.
SARS.
But she's talking specifically about this paper. Her Nobel Prize lecture is on YouTube. There's now basically, the new age is now this fantastic, unbelievable detections of amplification-free CRISPR. I'm going to define or talk here in a second about a new non-dimensional number that I'm proposing to evaluate some of this literature. But now it's very common. So because there's 1,000 papers a year and cumulatively maybe 5 or 6 or 10, maybe approaching 10,000 papers, there's now reviews, including reviews that are just to the fantastic papers. They're just reviews of attomolar type concentration, amplification-free, basically. Amplification-free detection is now a thing. So here's one more, and I'll kind of end with this, but you can imagine that in the absence of target, you measure a velocity of the reaction. Based on the Michaelis-Menten theory, if you scale that measured background, that's what BG here is, that measured background, by kcat, you get a pseudo concentration. This is the concentration that you would incorrectly assume is there for the target.
Yeah, if you got it.
If it were there, but it's actually coming from the background. And then you can divide that by the amount you reported. And the amounts that people are reporting are 1 million times larger than the background. So that's what this is sort of showing, that people are reporting about signals that are 1 million times smaller than background. We estimate it would take your little enzyme 27 years to produce a signal that you claim to detect. I don't know if that helps people. But. So to summarize, we really try to make an impact, try to make this field more quantitative, have people respect self-consistency, and it didn't work out. Most papers still published, and the biggest trend in kinetics is now they don't give you enough information, and then they refuse to share it. But even more worrisome is this, that we think almost all of these, at least 99% of these, are completely impossible. And that rate is just growing. So, I don't know. With that, that depressing thought, I'll leave you, yeah. Please.
Okay, now, questions?
I have a question.
Yeah, Juan, do you hear me?
Oh, yeah. Guys, hold on. Josh first, and then whoever just spoke. Okay.
Okay.
Is there any advice?
And then he's just 1 second. We'll come to you in a second. Okay.
Is there any advice that you would give to an incoming head of NIH or other federal agency to that would cut down, you think might help cut down on federal funding going to fraudulent science like this and perhaps much more? One of the things I thought when I was listening to your talk was, is this just the tip of the iceberg? Fortunately, we have Juan here who is able to detect this, but what else out there is, what other bodies of research out there are basically fraudulent? So interested if you have any thoughts on what the role of public policy might be to try to make sure that money isn't going to support fraudulent science. That's my question.
Yeah, no, that's a huge question, and I'm not sure what the answer is. I will say that I think the major failure here was editors. I think people all over the world, very well-meaning students all over the world, were finding kinetic rates that were 1,000 times lower than every other paper in the field. And it may be that in some cases, a PI told them, we'll keep doing it until you match the Nobel Prize winner, right? But at least some of those should have trusted and worked with their student enough to submit it. And what we found is at the editorial stage, it just gets shut down. So somehow I think there should be consequences for editors, even more than authors. You can't control authors and people's careers all over the world and what's now 1,000 different labs. There should be, the editor should be held accountable. That's what I think. I don't know how a government helps do this, but I think it's more the responsibility of scientists to do this.
Okay, so Annalise.
Yes, hi, you guys hear me?
Yes.
Okay, so Juan, you know, the only thing, this is so cool, really, I'm so glad I listened, and of course the only sad thing is that there's only 16 people on Zoom. I don't know how many are in the room, but too small a number. But in the spring, I'll be teaching a class called Advances in Biotechnology that usually has about 100 PhD students and master's students that all are interested in biotechnology. So I hope you'd be willing to reprise this lecture for that group of young scientists and explain all this, because this is so cool. I mean, it's sad, but I mean, it just shows you what can happen. And you know, I was thinking about, you know, that you were saying that you thought it was honest errors that led to these numbers, at least for those two papers. But you know, the thing is, was it or was it because they thought they knew the answer and so they just kind of picked the conditions that put them in the right order of magnitude, which was really the wrong order of magnitude?
Maybe I didn't express it right, but I'm happy to extend the benefit of a doubt to the first two papers.
Sure.
A heady time, those two are in very direct competition, this Berkeley and MIT group famously. You saw that court case. The second one sort of could rely on the first one as saying, oh, we get about the same numbers, so now we're done. There's certainly, but the following dozens of papers, I think they have no excuse. And none of those have published corrections, none of them.
Yeah, and all of the editors, pretty much so far, all of the editors and the DARPA program managers, with the exception of your own, they're just being cowards, you know?
Yeah, editors don't, I'm an editor of a Cambridge University press journal, founding, editor-in-chief. Editors feel a lot of pressure to not have retractions. And many things that should be handled as retractions are handled as core agenda, as corrections. So I can see the pressure, but it's no excuse. I would be happy to retract any paper shown to be completely impossible in our journal. Yeah.
Yeah.
Eric.
Two thoughts. One, for reform, I wonder why the NIH doesn't require that data be publicly available and the code used in a project, what you might think of that plan. The other, on what Annalise was talking about, is it looks like this might be good for somebody to write a paper on the file drawer problem or publication bias. So you have something that can't be correct empirical finding, but there's always measurement error. Could we figure out how many studies must have been done that never got published because they had boring results, but these got over the threshold accidentally? So,
Yeah, I think John's a world expert on this. I need he's here, but I'll try to say that. And tell me if you agree, John. But there's an inherent bias to getting really good result. If you don't get it, you may not write it up. You may not submit it. And then there's an inherent bias in favor of sloppy people. Sloppy people who get an incorrect, amazing, fantastic result will now write it up. And so they'll submit it. Some of these same people submit a proposal, and they're proposing something fantastic as well. And it's supposed to be that editors and reviewers hold the line, but some of these journals have so many papers, the editors handle so many papers. It's a time commitment, and apparently there's no repercussion. So I don't think we can fix it here today. Yeah.
We're going in order here. The next person is thanks. Right.
Very interesting. I was trying to go in the direction where Josh was going, but then I sort of, so I'm thinking, I really liked your answer about the editors, and I think what you meant to be is editor retraction policy, because of course the editor cannot know whether the original paper was wrong, right? But they should know.
And enforcement of their policy to share raw data.
Right.
That is not enforced.
Yeah, right. But then the question is, to what extent this is an example, because, there is also, there are some venture capitalists and some firms behind that. The consequence of, they're faking till they make it. So it's like, some of these firms were thinking about doing editing, but in reality, they want to sell something quickly. And so they BS on some of these dimensions, and they have an incentives to be supported by these publications. So, that the thing that I find interesting is the interaction between money, real money, like, commercial money, and science. I think it's great, when it works, but then the question is, the incentives could be really negative, right? Because you have some LPs probably that, gave 2 billion, to what extent they want to come out and say, oh, we found out there is a huge mistake in the original paper.
Maybe the most famous example is the Theranos, right? That was exactly a sensitivity issue. She and her company, Elizabeth Holmes, claimed a sensitivity from, 10 microliters of blood. That's basically impossible. Some of us very publicly stated, hey, this is impossible. But she was very popular. She went to Obama's White House and was,
But she had a
It's like a
board
big-
that was made of incompetent
She had a-
non-scientists,
exactly.
right? She chose it deliberately. She was very smart about that, right?
Yeah, it's amazing how much money you can raise without competent people checking.
Like, I remember I had a venture capitalist commenting on her when she was high, an app and says, look at the board. This board is very suspicious.
Yeah.
I don't trust the product.
Yeah. There's like ex-military people on
Right.
the board right now. And Stanford
Okay.
people, Stanford people
So-
are big.
I was,
Yeah, Schultz was there, yeah. Like, he was
I was-
also old.
sorry.
Oh, go ahead, we can hear you.
Okay, no, I thought you were continuing the conversation. I was going to suggest that this is very interesting to lay people. I think the biology as you presented it is maybe a little too difficult, but if you could get this written up in a form that someone like the Free Press might be interested in publishing, I think that's an interesting way to get the word out. I would go these other modern routes. To try to get people to hear it. I wrote recently a critical appraisal of the back to sleep literature, the idea of putting your babies to sleep on their backs as to prevent sudden infant death syndrome. That's a field where everyone comes up with the right answer. Everyone, even when it's not plausible. And that's how you get a quick publication. You get funding from the NIH. They want you to come up with this answer because they have a policy about it. You write the paper and you're done. But I think that this is sort of endemic in all of science. And I agree with Josh that the government is somehow involved in the sense that funding comes from the government and that some people in those positions have a sort of predetermined answer that they like. But just to encourage you to go to the Free Press or Sensible Medicine or someone else that has an audience like that.
So we had interviews when we published this, started putting online, and when our paper came out, when the corrections came out, we were contacted by reporters from CRISPR.com, from, what is this one? Genome Web, IEEE Spectrum. We did at least 4 reports. This is during COVID, so they were all over Zoom. One interview with the reporter took 3 hours. One of them wrote a couple thousand words, and we very meticulously reviewed it, edited. All 4 stories and all 4 interviews were spiked, not a thing published.
Yeah,
As soon as it went to the IEEE Spectrum editor, he said, we're not publishing this.
I wonder why.
So I put up videos of me giving this talk, or the first part of the talk before this worst problem, which is the attomolar. Before the attomolar stuff, the stuff about the incorrect kinetic rates, I put it on YouTube, we put it on our website. We published 7 papers. I published a perspective where there's very little equations. There's 3 algebraic equations. They're really ratios. You just need to divide 2 numbers. Okay. In economy, you guys multiply and divide. In economics, right? You're good at both.
Yes, and sometimes you get the right answer.
Yes. Yeah. Subtraction, addition's tricky, pal. Anyway, very simple things. And it doesn't really help. Some people really get it. There's people out there that cite us. Here's just a funny one. There's a CRISPR memes Twitter, X, where they posted this, and there's a lot of nice comments on it. But, you know, this is us here. This is Ashwin and me knocking on doors of seminal paper with CRISPR.
That's cute.
So people get it. People are published alpha, beta, gamma. I would say like 10% of the reviews recognize that there was this catastrophe, and they cite us. Two of my papers are the second and third most cited in this CRISPR stuff. So all that sounds great, right? But in the end, we only affect about less than 10% of the papers, maybe 8% of the papers recognizes. The other 92% are still publishing garbage.
Okay, Ivan.
Hey, Jonathan. Hey, Juan. How are you? Sorry I cannot be there. Very, very nice presentation. I found your results quite shocking, actually. And you talk about scientific misconduct. But the thing I find sort of hard to believe is sort of the scale of what you're describing. Because I can see, 2, 3, 5 people, maybe even 10 people, especially those who have economic incentives, sort of manipulating at that level the evidence. But what is really hard for me to believe is you're going to have hundreds or even thousands of people making huge mistakes. There are not even sort of statistical mistakes. You're talking about internal consistency. It's hard to believe that there is something of that scale in other sciences. You guys correct me if I'm wrong, but I feel like, we have evidence of these things in economics and other social sciences. But typically what you would see maybe is people sort of manipulating doing some p-hacking on the margin, but that would be something maybe to the tune of maybe 1-2% of the magnitude of the results. So I find it shocking and quite depressing, because we tend to think that the scientific process works, right? Yeah, some people can make mistakes sometimes, but this sort of egregious mistake being done almost by the entire field, right, is something that I think is quite unprecedented. And I guess the question is there something special about this field, and is it potentially also a case of huge incompetence more than scientific misconduct? Sorry for the long question.
Yeah, no, that's a great point. To explain it with incompetence, you'd have to maybe say that all the bias is because if people misrepresent it 1,000 times lower, the right value, or 1,000 times higher, then there's some sort of bias in selection for the 1,000 times higher. So I don't think incompetence can do it. All the mistakes are always in their favor, and they're 1,000-fold mistakes. So I think incompetence is hard. That's why I thought about it a lot, but I did put misbehavior, or whatever the title, misconduct. I can't explain it any other way. If I'm wrong, I'd love to see the evidence.
It's just that it seems almost suicidal to do it at this point, right? If you continue
So actually, the other interesting trend, and I just put this up here, these are reviews of the papers which are almost all impossible. It's this amplification-free 1 attomolar results. So for example, 1 attomolar results, where the real number, just because of the background activity of the enzyme and the imperfections of the reporters, give you signals that are 1,000,000 times greater than this, right? So with no target in the room, you get these numbers. The other interesting thing is China. Something like of the ones in this slide, 90% of these fantastic papers are coming out of China. That's another pattern I see.
Okay.
And in good journals, in Western journals, and they come out of China. Yeah.
So trying to get.
Wait, hold on. Over here.
So nominally, these things are being used then to create companies that are going to put out these products that people will use, right? So is it that the companies just don't care that what they're offering doesn't work? Or that will eventually be found and the companies will collapse?
Eventually they found it. So we saw, for example, Mammoth,
Yes.
laid off like 34 scientists.
Right.
From one day to the next. So eventually the rubber hits the road, and if you put it out, or maybe it would be caught by the FDA, where you have to have double-blind tests, and then you'd have a bunch of false.
So eventually the whole thing will collapse.
It just gets swept under the rug. It doesn't collapse. They just change their story. So now they're gonna have to do the same thing, but with amplification. And the consumer doesn't really care because they don't care about amplification or kinetic rates. I think more accurate is to say the whole thing is sort of swept under the rug. Let's pretend it never happened, but yeah, we have to go on. So if you're publishing, you still don't need amplification. If you're selling instruments, yeah, you have to have amplification. And that's it. That's a very nice compromise. Very clear. Everybody's happy. Yeah.
Okay, I'm gonna want to say something, but these two, and then go, you first.
How much retaliation, such as loss of research funds, have you encountered because of this?
I love that question. I've submitted 6 or 7 proposals on CRISPR and not one has been funded.
But have you lost fund? You seem to have money.
No. Ashwin had a fellowship from BioX. These are all side projects of students where the money comes from something else.
But
I don't work
so
on
you have money, just this is you're siphoning off other projects to fund this.
Oh, yeah.
So you have money.
I have students with fellowships.
Yeah, okay.
Which I think is essential to the future of this university. With the union and the rising cost of grad students, we have to have fellowships to do real good work. So that our development office should be out raising fellowships for researchers. But anyway, that's a little parochial thing.
I'm trying to put this into perspective. These 1,000 maybe plus papers
Yeah.
that have implausible results.
Yeah.
Are these implausible results like 100% of the paper construct, or is it like they have this, but they have another 10 parts in the paper, and maybe the other 9 are okay, or maybe they're also horrible and implausible?
I see.
And the second question is, trying to reverse engineer, what do you think happens in these labs? So is it fake, fraud? And if so, how exactly does it work? Is it lack of supervision? Is it like some sort of copy paste, some negligence? I mean, can you imagine what is going on?
So to answer your first question, the way I'm counting these is with Web of Science and Google Scholar searches, where they specifically tout amplification-free CRISPR in their abstract or title. So they're really leading with this. And I've spot checked them. I haven't read the 1,000, but I've spot checked 30 or 40, getting ready for this talk specifically. And maybe of the 40 I read, all 40 made this a big deal. Right?
So it's a key topic.
They're basically saying we now have a way to do this without labeling, because maybe they do electrochemical detection or Raman scatter. It's now like the time when all the sensor people come into it. And the sensor people can always get signal out of something.
So it could be that there's many more papers where this is not like.
Oh, for sure.
It could be like 20.
Exactly. There could be many more papers. That's a great point. Yeah, there could be many more papers that I didn't find because I'm only looking for those that highlighted.
Yeah.
The second question, I can just pose hypotheses, but I think there's lack of courage. I blame PIs. If you're a student, say from India or China in the US, and you measure a kinetic rate, you go to your advisor, as mine did, and say, I get this value, it's 1,000 times less, right? And I'm guilty of telling Ashwin, well, how can you get a number 1,000 times lower than everybody else gets? You know, there's person has a Nobel Prize. Want me to show you the YouTube video? So Ashwin kept doing it again. Ashwin is an extraordinary person, incredibly honest, scientifically honest, and courageous young man. And I ended up going into the lab with him. He would run the number with me. He would show me all the calculations. And then I'm like, okay, let's try to do it in a more simple manner. What's the one piece of raw data where we can make a very simple calculation, just a small, like the time scale is a big one. If I ignore your y-axis, I need your time scale to start turning over by this value. And then how do you know that concentration is what you think it is? And then we went through all of it. I think most PIs don't do that. I think most PIs just say, it's too low, keep repeating it. And there's at least some people who want to maybe keep their visa. They want to graduate. And some people feel like their job is to find the right answer. Yeah.
So Juan, let me give you a different perspective on this, which is that what you have here is a tremendously good experiment because you've got the violation of energy conservation on your hand.
Mass conservation.
So, but now let's take a step back. I mean, in economics, we don't have these fundamentally. What happens in economics ubiquitously is somebody will do something stupid. Somebody else will repeat the stupid mistake. And before you know it, we have a literature all doing that. And if you question it, they say, oh no, I'm just doing what this previous person did. That is unbelievably common, right? And we're not talking about violation of mass. It's be something that's kind of minor, but maybe not, who knows? But we don't have the ability to say, look, this is wrong in this very strong sense that you have that ability to say. But I think what you see happens afterwards then occurs. So you have 1,000 papers on the subject, right? So you have an entire literature in economics on the subject that now you can't write that correction. And the half-life is enormously long, right? So I think what you have here is not so much a misconduct thing, which it is a misconduct, although the original authors corrected.
Yeah.
So I think in that sense, it's,
very appropriate.
yeah. But where I think what you have is how, because of the incentives, so you say it's the PI's incentive, right? Well, sir, I don't have a PI. I don't have a runner lab. I don't have a PI. But the average economist is saying, look, I don't want to put the effort in to correct this. That's not going to get me anywhere in life. I'm much better off doing what everybody else did because I'm going on to something else.
You do have an editor, and you have your own bias and you have your editor's bias. And those, I think, are the biggest ones.
But the editors just give the same.
Sorry?
What about career concern in general? Because I'm with Jonathan on this, and tenure review. Like, just this is one thing that is actually big for us as well, like, especially on the replication, and let's talk about maybe organizational behavioral cases, right? So you have all this lab experiment, but you don't know how these were done, right? Especially the early one. It would be nice with these, following what Jonathan was saying, to just study that requires a bunch of people looking at, were these tenure people, were just trying to graduate, and, and maybe the career concern is likely different because I would say that 50% of the people don't even go in academia.
But what I'm saying is it's just what do you want to call it, a meme or something? I mean, I had this experience with you, Josh, when we worked on that paper together. And I went to the labor literature and they claim something. Look, I can make mistakes. But every single paper, and I'm talking thousands, started from this one paper that made a claim, a mathematical claim, not a data claim, that I, to this day, think is incorrect. Now, what is my review at this point? I'm a financial economist, I'm a labor economist, what do I care about this? Am I really going to sit there and say, what they do in economics often is give names to stuff. that don't correspond to what is actually going on, right? And then everybody, you cause it, and then it just explodes. So now what's my incentive to do something at this point? My incentive to say, Josh, I don't want to do this. I don't want to get involved in this. I don't want to, and so I step away. And I think that's what happens when people from outside the area look at something and they see something that's violated, intellectually violated, they just say, I don't want to get involved in this. Let this area completely continue to do what they're doing.
One hypothesis is anything that's hot and really growing,
Yeah.
there's something wrong there. There's something wrong there.
But it may not be, it may not be, because you have a confluence in this case of a Nobel Prize granted pretty much at the same time, so nobody wants to cross those people. So it may not be that.
Reputation is a big one.
Yeah.
These two people have the biggest and have the biggest reputation in the entire field. And they really saved their reputation by
Correct.
publishing a correction, yeah. But the other, hundreds of papers.
They didn't read.
Was that Nobel Prize awarded directly for amplification-free CRISPR?
No. Most of it is the gene editing,
Gene editing.
which is extremely well-deserved, but proven in court, or Zhang would argue, the Broad Institute would argue, actually, Feng Zhang did that. But
But what's interesting is you could actually see that, right? The fact that the numbers were both 1,000 suggests to me there was some communication. So you could see whose number went. If you could somehow see that, you'd see exactly who copied who.
They set a standard, and you had to meet the standard. I mean, this is a fast.
No, but they
future
both
that
came up with the same number. That's too coincidental.
Yeah.
They both made
The same number ordered to them.
So lunch, who wants to go for lunch?
I could